Welcome to the hands-on session. This is day 8 of the workshop. In this hands-on session, participants will learn about network analysis. Omics research throws up a lot of data which is very hard to interpret using traditional methods. Network analysis is a crucial tool for analyzing the high-throughput proteomics data. In network analysis, we evaluate how nodes relate to one another to provide a structure that helps reduce the complexity of underlying data. Using network analysis tool, one can easily interpret the biology behind those extensive datasets that mass spectrometry-based studies usually generate. This session will be a hands-on session on network analysis tool, namely Metascape. We hope this session will be a learning experience for all of you. Thank you.
All right, everyone. So this session, primarily, we will try to have more time where you can see some demonstration by Dr. Arup to show you how to analyze your data and make the networks from that. But before we come to the hands-on part, I'd like to continue where I stopped the previous lecture on the protein microarrays, and just give you some idea for the workflow involved in doing these type of protein array-based work. And then we will come to the data analysis part. So as we were discussing in the morning quite interactive session, that there are different type of array-based approaches which are possible. But questions are, how to make these good arrays and what are the considerations for doing these type of experiment, which many of you are not very familiar with, right? And I'd like to also give you some brief overview of autoantibody-based screening protocol. So first question is, these arrays which you see, some beautiful red and green colors with some white and yellow patches in between. Those come with a lot of hard work, with making these good arrays are really a very important science and art combination, which brings these kind of arrays. And that's where array-based technologies, they're still quite limited because, there are many people cannot make very good arrays. So there are some very expert companies and the scientists who are really dedicated for doing this technology. If you are planning to do, let's say, protein array-based work, as I mentioned, unlike the mass spec-based work, where you do not need to start with a hypothesis, you can take the samples, extract protein, and run them the shotgun approach, and build the hypothesis based on what data you get. In this case here, you are starting with designing a microarray chip first, and making experimental plan based on that. So you have to be very much thinking about what type of experiments are you going to do, because accordingly, the type of clones which you are going to print on the chip, or the controls which you are going to have, positive and negative controls, all of those are going to be part of your array layout. So those has to be immediately thought of. Then which type of substrate you are going to use, what type of surface chemistry you are going to use, how you are going to print your peptides or proteins, or if you are planning to look for the cell-free expression arrays, then you are probably planning to print the DNA. How are you going to print those? So those are the kind of things which you have to think ahead of time. If you are intending to work on the cell-free expression arrays, you are going to start with culturing your construct clones. Those will have some tags which you have to keep in mind, because accordingly your array chemistry will work. You're going to purify and concentrate the DNA, resuspend them in the right type of spotting buffer. And once you're ready with the mixture, either going with this route of cell-free expression based on the DNA-based work, or taking the proteins directly or peptide, then you have to do the printing part. That is the next part, array printing. And how good your printing has come, and are you ready to perform your next set of experiment, that comes under the quality control check. So all of these are important considerations, and I'll just give you some glimpses in the process so that you are aware that how things actually work. Now let's say you want to print a small array yourself, and I'll show you maybe some hands-on in the post-lunch session how some of these arrays can be printed. There are some printing head in which the pins are there, which is going to be used to print these arrays. So you make the layout like that, that how many pins you have for printing your arrays. So let's say in this case we have 48 pin microarray head. And then what is the array layout pattern which you're going to make? You decide those, and then only you feed this information in the software. Then software is going to pick that layout. Accordingly, is going to dispense the content and print the contents on the microscopic glass slides. So these are something which you have to think, and while printing, you have to make sure that you maintain the humidity properly, temperature properly, make the deck with the vacuum, how many pins you want to use for printing, their numbers, what type of, if you are going to take 96 well plates from the glycerol stock followed by protein purification or DNA, or you're going to take 384 well plates, those has to be decided. Number of slides to be printed, sometimes 30 slides, sometimes 180 slides, sometimes even 300 slides in single shot can be printed. What type of array mapping you're going to use, the densities of the arrays, where the spots are going to be printed, and of course many printing parameters, the inking, stamping, print depth, how to wash the your pens during the printing. So you can think about making good arrays are quite a bit steps are involved, and that's where there are limited vendors and commercial companies who are offering these good services, and they are actually quite costly in that way. So if you are, let's say, planning to make a small array yourself, and that's what I think, my this part should make you familiar that can you do small arrays yourself for the your validation experiments or to take your leads forward for the clinical translation value. So now on one of this slide, you want to print many features, right? So now let's say this is the one of the block, which is showing you the layout of 6 by 6. How many features you want to print on this particular one block, and you got so many blocks on the one slide. So here what you see, the inset of one of this particular feature here is that you can print so many genes here. And how you want is that you don't want the same proteins to be printed next to each other, or the DNA, so that you have no neighboring effect. You want to have the duplicates for the reproducibility, like 11 is across corner and 8 is in the corner diagonal. And this is something which you should keep in mind. Your duplicates should not be next to each other. Now, if you're looking at IgG response, immunoglobulin, for doing the immunoassays on the chip, you should be thinking about printing some purified IgG with the dilution series of, from lower to higher concentration. And those will light up, which can help you to decide that what was the immune response, which can be compared with the proteins expressed on the chip. And these are some of the real experimental slides which I'm showing you here. In this case, you're looking at some of the breast cancer patients, and one of the highly expressing protein, this is the EBNA protein for the breast cancer, was very highly expressing, and all the neighboring spots are really getting, affected because of the huge expression of this protein. So that is something you have to keep in mind. Maybe in the next set of experiment, you have to keep this protein separately and avoid with the so much close neighbored spots for these activities. You want to look at different spots, their expression values, their duplicates are looking good. So these things have to be performed in the protein arrays quite a bit, and especially when you are in the early stage of development of the protein chips, you have to look at the spot size and morphology. They should not look like this kind of waves and the diffused spots, or this kind of bad printing, or this is a donut type of stuff is coming with the printing, which should not be the case. You are looking for this kind of ideal spot morphology, which is quite consistent spot to spot. Then only these arrays can be taken forward for your actual biomarker based experiments. So you are looking for good quality chips like these, and you have to keep many parameters in mind before you actually proceed for your the real biological experiment. All of these steps are not possible without liquid handlers. So this entire making the arrays and performing these arrays based experiment requires lot of automation and robotic power, as well as liquid handlers. Strict sample tracking has to be done in the entire process, because you are talking about thousands of proteins or DNA to be printed, and then going to screen hundreds of samples. So a lot of barcoded based things should be in place, so that you can easily access the plates, and you cannot screw up with the labeling. So careful quality control of materials are required. detailed description should be mentioned, and of course you have to make sure that you are following the same protocol again and again, and there is not much variability which you find in different batches of the printing. So you print one batch this month and another batch of the slides 2 months later. You have to keep comparing those batches as well. So spot to spot variability, for instance, duplicate spot I showed you, slide to slide variability, like you are printing 90 slides in a batch. The first slide and 90th slide, what is the variability in those slides, and day-to-day variability when you do the same experiment today or after a week, how much day-to-day they are close or not. So many of these things you should do these plots to find out, these things are really showing the very tight CVs, and you're actually good to go for the actual biological experiment. So I hope you're getting idea that before you start your first biological experiment, lot of things actually has to be looked into the quality control parameters. I spoke to you briefly about nucleic acid programmed protein arrays in the first lecture. So the typically one of this experiment involves that you have printed your DNA of interest on the chip, and this is an Napa array. Now you're going to block this array with the blocking solution, which is BSA with the milk, or you have the super block solution. You block so that all the empty surface of the slide that is blocked, and there is not going to have the too much background from that. So then now you're going to move to the IVT mix addition, in vitro transcription translation mix, which you're adding. And you are after addition of that, you are going to incubate that at 30 degrees for 90 minutes, followed by a capture step, which is 50 degrees for 30 minutes. Why it does that? Because I showed you there's a capture antibody there. So if, first of all, you want to express the protein, for which 30 degrees is a good temperature to do that part, and then you want some time for this express protein to capture and bind to the antibody, capture antibody, and lowering temperature 15 degrees helps to do that binding. So in 2 hours from the DNA, you are now having the proteins on the array, and that is getting expressed. After this step, again you are going to block your chip. Now you are ready for starting your experiment. If in this case you are going to look for the patient serum sample, you can incubate with that. Or so if you're looking for autoantibody based experiment, your hypothesis is that antibodies are generated in the patient serum sample. So that could be used as the sample with the primary antibody, or you can actually take a primary antibody separately, and you can incubate that, whatever your total design is about for that project. Then you can again incubate for around 45 minutes or so, add secondary antibody, and then followed by the signal detection system. In this case, I've showed you TSA, tyramide signal amplification system, but there are different type of peroxidase based and other type of assays can be used to detect your signal. So broadly, it is very much like Western blot. If you see, like, the steps are very similar to the Western blot. You have to make sure that background is not very high, there is no non-specific binding is coming, and you are able to get good signal over background, right? So these are the kind of thing which you are going to do. In this case, there is an innovation here where you are starting with the DNA with the tags, and along with those, the capture antibody is printed on each feature. Then after doing these expression, you are going to make the protein. Protein is the binding with the the capture antibody, and then rest of the steps are like the Western blot. You can play with this place here to look for what is your experimental design. You are looking at a specific response of a given protein. You are looking at protein interaction. You are looking at the patient serum sample without antibody response. This is the place when you can change that variable. So when we do this kind of experiment with the Napa, for example, you want to first make sure that your DNA printing is good, and also you want to make sure that protein is actually getting expressed. Both things you have to make sure. So in this case, you're going to use anti-GST antibody, because every protein which is expressed is having GST tag. So and there are many gaps here as well, which is actually empty spots in other regions. So you are going to look for the protein expression using the anti-GST, let's say in this case, although you can use different tags as well, as I mentioned earlier. And then the next slides show that you can also look for a specific protein response. For instance, on this big slide, there are 2 spots of P53. So if I use anti-P53 antibody, they are only showing up here. So it means all the proteins are getting expressed, and protein expression is quite unique and very specific as well, because we can actually see specific protein expression for this particular protein on the array, and nothing else is lighting up on that. So ideally for doing this experiment or before going to proceed for your biological experiment, when you make these arrays, first thing is you want to find out the quality of the printing of the chip. You're looking at the quality of the DNA printing. You can do the picogreen staining to look, just monitor the DNA printing. You can now look for the protein expression but with the anti-GST antibody. You can look at specific protein expression with any antibodies of interest. And after doing these QC checks, now you are happy that, okay, now these arrays are good for next set of experiments. And then, of course, you are going to keep comparing different batches of the printing as you go along in your experiments, right? So now you got to feel that doing good microarray experiment is also going to require you a lot of considerations, like what we have been speaking in the mass spec part. Here, there are a lot more things which you have to keep in mind. So the simple experimental plan for doing autoantibody screening based experiments for different autoimmune disorders or even different cancer people have explored to screen on these chips. As I said earlier, that you will take the chip, block that with a super block on the room temperature for almost an hour or so, or you can do it 4 degrees. After that, you're going to add the serum sample, and again many optimizations are required, but in our experience with this dilution 1:500 serum dilution, that worked well. But that is no set guideline. You can always optimize what serum sample you have from which type of disease, which type of patient samples. You have to adjust the dilution so that there's not too much background. So I cannot give you exact number. It can be directly undiluted. It could also be 1:200 dilution or 1:1000 dilution, right? Then you incubate for an hour or so at 37 degrees. After that, you do the washing part. You add the secondary antibody for detecting the signal. In this case, the Psi labeled. Scan the slides and you can obtain the images for analysis. There are many considerations while doing these steps. You have to do the printing. We will show briefly about these things in the afternoon. You are going to look for how many pins you can have, and accordingly your array quality can be checked out later on. Even during the printing, if you're making your own arrays, you have to look for variety of details, how many pins you have, the array designs, how much time you're giving for dipping, how much time you're giving for cleaning the pins. All of these steps are to be set up yourself. If you are buying the commercial chip, of course you don't have to worry for these stage and these steps. If you are not going to be actively using it, probably you're going to rely on the commercial facility or collaborators who can do that. But even when you get the chips, now you can start your experiments. You will have to have these kind of beaten trays where you can place your arrays in these trays and do the blocking step or washing steps. And once you have done these experiments, then you are going to play with the scanners, which we will again show you in the post-lunch, and play with the various parameter of scanning, especially PMT gains, and look for how good the slide is coming and looking the images. Place the array grid on that, lay the grid on the chip, and then you can look for all the detail of the protein which is printed with the GAL file, which is a part of this layout. These are some of the images which we are showing. Some of them are not good here. We want to illustrate that when you are scanning the slide, you should be paying attention to how good the slide is looking. This is one of the very high density human proteome array we are talking. And this slide definitely shows with that, if you look at protein expression, they're not very uniform. So this may not be best slide to take forward. And when you do the immune response screening, again, sometime you find in this case, probably the patient serum was too much concentrated. Dilution was not appropriate, too much background, we can see it here. So these regions may not be good actually to take forward. So you need to optimize this slide. Either this could be array problem or this could be the serum concentration problem. You have to actually avoid these background issues, right? So typically, if everything goes well, from a patient's serum sample with very few microliters, which you can dilute in the buffer, you can put those into the microarrays, and potentially you will be able to identify the signature of the protein which is binding to these features here, which is showing the antibody response. And along with this, of course, many other applications are also possible on the arrays. People also look for protein-protein interaction, protein other biomolecules, as well as different type of PTM analysis. For those type of PTM analysis, as we briefly discussed, you have to provide the right conditions for the phosphorylation to happen. So you have to add those kinases and ATP. For the glycosylation to happen, you have to add the machinery which can do glycosylation, especially with the rabbit reticulocyte lysate, there is already possibility of having glycosylation, but you need to do those experiments separately, whether the assay condition is favoring the PTM modifications or not. And if that is the case, then you can apply the same on the miniaturized array format. So I hope you're at least gaining some more familiarity of using a new technology which can be utilized for your different projects for protein array-based applications. Just to summarize the morning and this lecture, that different type of arrays are possible. Purified protein-based arrays, cell-based system, or cell-free systems. This is definitely one of the strong platforms for looking at high throughput ways of protein interactions or biomarker discovery based projects. However, it is not very straightforward to just take an experiment and try to obtain successful results from that. You have to be very careful in terms of the experimental design, what kind of sample you're going to screen for, what is the array layout, what are the controls you're going to use. And once you have identified these, like any other technology, then still you need to go forward to the next set of experiment for validating your findings. In fact, the large biomarker discovery programs, they start with, let's say, we want to work on one of the cancer type and want to look for autoantibody response. So you start with, let's say, 200 chips, and those can be used for the discovery set. When you are now looking at 30,000 spots printed on the arrays, you are looking for all the 200 patient samples to be screened. After doing the entire screening and analysis, you have found some proteins which are showing good response in different grade-wise comparison. Now you can narrow down those proteins, and you can print another array now. Rather than having 30,000 proteins now printed, you can now maybe print 500 spots, which is of your interest. And now you can take some of the same sample which you have used from 200, maybe you can take 50 samples from that, and you can try to see is that making the same pattern. And also you can take 50 more new sample which can be used for validation of that, and then that could serve as a validation strategy, a validation set of the array. After doing these, you can again narrow down your leads, and now you want to make a more miniaturized array, and now you can apply that with a blinded set of samples. So now you do not know what is the control, what is the cancer, what is the grade type. You are taking all those 200, 400 samples in the blinded manner, and now you are going to apply those on the final chip, and you are going to look at the response. So now you think about a biomarker discovery program, how that progresses from the really big experiments and the exploratory experiment, narrowing it down, validating those, and then reaching to the blinded state. Then only if you have reached there, you can then either simply make the ELISA type of test from those, or make miniaturized chips which can be clinically applicable for testing. I think that's all for this brief lecture session from this part of the arrays. Now we are going to have some data analysis part of taking any data set of proteomics, not necessarily only the protein arrays, but protein arrays or mass spec, they're both going to provide you big data sets, which has to be analyzed to make biological sense of that data. And that's where different softwares and open access things are very handy, which provides you biological insight of your data. Metascape is one of those. And now I request Dr. Arup from my team to give a demo of this part, and then we would request all the participants to perform the same experiments on the file which we are providing to you. That's all from my side. Thank you, everybody. Arup, please take over.
Thank you, sir. Right. So before we start using Metascape, let us just understand what is network analysis. If my screen is visible.
Yes, now it is coming. Yeah, make it full screen.
Right. So
please make it full screen. Yeah, now. Go ahead.
Right. So for network analysis or pathway analysis, so the problem is whenever we start an experiment is at the end of the experiment we have lots of data. These are all high throughput techniques. So humanly it is not possible to extract all the information that the instrument has given to us. So it often happens that you would have a lot of hits. You would have 3,000 or 4,000 proteins. But you really do not know what these proteins mean. I mean, why is a particular gene or protein? I would use these terms interchangeably. So why is it suddenly there are so many genes getting upregulated and downregulated? So we use a term called pathway enrichment studies, where let me show my pointer. So a traditional approach would be to go for take up a favorite gene, use it in PubMed and see how many papers are published. And then you try to make sense of the data. But suppose you have 3,000 hits. You would spend an entire year just to understand your data. And science moves pretty fast. We cannot use those techniques. So traditional approaches are not viable. So what we need is intervention from technology. So what pathway and network analysis tools allows us to do is gives us a mechanistic insight into the omics data. And it helps us to interpret our results. It just helps us. It does not interpret the results for us. So as a scientist, our duty is still there, that we have to look for that what was our biological question. And then we have to use these tools judiciously. It's not that you will put in a data set and next day you would have a paper in Nature. So that does not happen like that. So just before we begin, so there is a small difference between the pathway analysis and network analysis. Well, pathways are always very detailed, high confidence and consensus datasets. They include biochemical reaction. They are a bit small scale, so there are fewer genes, and they are the information is taken from literature. These are very robust datasets. Whereas network analysis are more AI driven, so there is lots of abstraction. It's noisy, and obviously it's constructed from omics data integration. So just remember that there's a basic difference between pathway enrichment studies and network analysis studies. Why do we do pathway analysis? Just as I said, it is easier to interpret. So once you have a set of 500 or 600 upregulated genes, you would like to know that what these genes does. So they would introduce you to familiar concept. So it may tell you that it is involved in mitosis, it is involved in cell cycle, it is involved in reproduction biology. So it makes it possible for you to identify causal mechanisms. You can also, it can help you to give new roles for a particular protein. It improves the statistical power of your analysis, and it makes your data more reproducible. So mass spectrometry based studies or any omics high throughput studies, one thing you would find is it's not extremely reproducible. So in the data set I have shared with you, which we will just go through in some time, you would see there sometimes with patient to patient variation is there. So when you're working with clinicians, the patient itself might vary. So there you need a protein expression signature. You might not need a particular, you just need a trend that, okay, this particular protein was present in more patients or highly overexpressed in more patients. So this signature studies, it helps you to do that. So if we talk of pathway network analysis types, there is an enrichment. There are three types. So one is the enrichment of fixed gene sets. Then there is a de novo subnetwork construction and clustering. And then there is pathway-based modeling. And every idea. So why do we use these three approaches? Because these three approaches actually help you answer three different types of questions. So if you are talking of enrichment of fixed gene sets, which we would be doing, so there we would get to know that what biological processes are altered in this cancer. Whereas if you do a de novo subnetwork construction and clustering, you would know, are new pathways altered in this cancer? Are there any clinically relevant tumor subtype? So you see, the biological question you're asking is changing. And then if you do a pathway-based modeling, the question that you would be asking is, how are pathway activities altered in a particular patient? So it takes you to more of a, precision medicine type of, these are druggable targets? Can we design a drug for this patient? So all pathway or network analysis, it starts with your biological questions. So it's very important to keep in mind that whenever you start your analysis or rather design your experiment, your biological questions should be very clear. So a pathway overview would be, first you collect the proteomics data. It come from a mass spec. Then as I think we have already explained to you in a previous session, you can normalize the score, compute the differential expression. All these things can be done on MetaboAnalyst and MaxQuant. Then you generate a gene list. Then you start using, learn the underlying cellular mechanism. Then you identify and visualize the networks. Then you try to understand the molecular mechanism. So this is where you start generating a hypothesis. Mostly when you are doing a mass spec experiment, these are the LFQ studies, mostly are discovery driven. You don't have a particular hypothesis. But these pathway analysis tools will help you to generate your own hypothesis. And then you can publish a paper that will explain the data. So before analysis, you should always remember to take these steps. The data file that you have got, the analysis part, these parts are already done. Because remember, for any analysis using softwares, there's a very important adage: garbage in, garbage out. You throw in garbage, the software gives you garbage in return. Okay? So the first one, as I say, biological question should be very important. And it should be part of your experimental design, because that will change the type of analysis you would want to do. So I'm just rushing through. Suppose this is a hypothetical work where, it's actually a work that we did in our lab. So we took blood samples from COVID-19 patients, and we wanted to know what happens. So the pathway observation told us that genes involved in blood coagulation are altered. So we did more statistical analysis, and we arrived at the hypothesis that blood coagulopathies increase in COVID-19. So any experiment you would be doing for your study, you would be also starting from a similar strategy. One more important thing that you really need to know while doing this is the gene and protein identifiers. So identifiers are like the Aadhaar number and the PAN number for you. So every gene or every protein will have its own identifier. And multiple databases have different types of identifier. Just like your Aadhaar number and PAN number is not same. Your voter card, I-card, and the driving license all identify you, but basically the numbers are not same. So you should use a similar identifier. If you're using UniProt, use UniProt. If you're using the entrance gene ID, use the entrance gene ID. Don't mix and match. So these are the common identifiers. You see, this is only for one single protein, but there are so many kinds of identifiers. So what we really need to keep in mind is that there are identifiers like the ensemble identifier, the RefSec identifier, UniProt. Basically, if you're using proteomics, we generally use a UniProt identifier. So don't mix and match. Because softwares can only identify a handful of the IDs. And if you use different IDs, you can, strange problems can crop up. Like if you talk of P53 gene, there all these four names identify the same gene. So let's stick to one particular. Another thing that you need to work, since you would be working with Excel. Now Excel is a very smart software. So sometimes your gene name might be converted into a date. Like if you write OCT4, it might be October 4. So always when you're handling the data, paste it as simple text. Another problem is coverage. Now all databases or all databases that we'll be discussing might not have your gene of interest mapped. So there will be a problem due to versions. Maybe the new version has it, the older version does not have it. So you have to manually annotate few of your proteins. We'll come to it very soon. So if, as I was telling, what's in a name? So you see there was a famous retraction from a Nature journal. So the retraction was of the paper HES1. What they actually wanted to say is homolog of ES1 and not from the gene encoding the transcriptional repressor hairy enhancer of split, which is also called HES1. So whenever you are studying or you are analyzing, since there will be so many of target proteins, maybe 100 or 500 or 600, it won't be possible for you to manually go and check it. Ideally you should, but sometimes if you want to skip that step altogether, please stick with ID that is quite universal, like UniProt. So as I told, species coverage, not all species are not equally represented. So if you talk with Rattus novagicus, it has the maximum number of experimental and non-experimental proteins covered. Then comes Homo sapiens. So it might happen when you are, so this is a pretty old data set. it might happen the species you are working with, it does not have all its protein IDs mapped. So there you have to use manually annotate, and you cannot automate all the steps. So coming to Metascape, now what Metascape does is it's a meta-analysis software. So you have multiple pathway enrichment tools, and an ideal condition would be that you take all these tools together and analyze a single data set. So the data set would give you a much more, a robust, holistic view. So Metascape does the same thing. So for pathway and process enrichment analysis, Metascape actually uses multiple gene sets. So there is a reactome gene set, there is a gene ontology biological process, there is wiki pathways, there is quorum, there is KEGG pathway. So although you're running a single analysis, what this software allows you to do is run parallel multiple analysis using the same data set. This allows you to get a better picture. I would briefly describe, so Reactome is a pathway database. We will be talking with the inventor or the man behind Reactome today. So I'm not dwelling deep into it. Then there is the gene ontology knowledge base. It's the largest source for information on function of genes. Then there is wiki pathways. Wiki pathways, just like Wikipedia, is crowdsourced, and there is a lot of curation as well. Then there is a quorum database. So this database is again a source of manually annotated protein complexes from mammalian organisms. So quorum is manually annotated, and the recent version contains 4,274 mammalian protein complexes built from 4,473 different gene sets. There's also the KEGG pathway, so Kyoto Encyclopedia of Genes and Genomes. So their pathways also have maps on metabolism, genetic information processing, environmental information processing, cellular processes, so on and so forth. Metascape also allows you to do protein-protein interaction and enrichment analysis. So we know that proteins do not work alone. They interact with other proteins. But wouldn't it be great if we put in our data and somebody tells us that out of the 400 expressed proteins, 70 of them forms a part of a protein-protein interaction network. So this is a very valuable information. And what Metascape does is it runs your information through BioGrid, that is the Biological General Repository for interaction datasets. Then it also does analysis on the string database. It also runs the same analysis on a protein-protein interaction network on the InWeb IM. And this is built by a team from Massachusetts General Hospital and the Broad Institute. Then there is a quality control and association studies. So quality control and association is how good your data matches with other data sets available publicly. So one is the trust. This is the transcriptional regulatory relationship unraveled by sentence-based text mining. So this is another manually curated database of transcriptional regulatory networks. Basically, it will tell you what are the transcription factors that can be involved in your particular your experimental data. How does it map with the transcription factors that are already present? So you get also understanding this, how this proteome is being regulated at the level of transcriptional regulation. Then there is OmniPath. So it's again a manually curated database. So it has protein-protein and gene regulatory interaction studies, enzyme PTM relationships. Then there is a human phenotype ontology database. So it is a phenotypic abnormality such as orphanet, decipher, online Mendelian inheritance in man, so on and so forth. There is also the DisGenNet. So it is a largest publicly available collection of genes and variants associated with human diseases. Then there is the pattern gene database. So it involves specific genes and selective housekeeping genes and repressed genes. So what the idea is, what Metascape allows you to do is it runs parallel enrichment pathways. Now we will just jump into a Metascape analysis. If all of you are having the files with you, I think you should check your WhatsApp and email.
Can you also place the link here in the chat box if it is in the Google Drive link? That would be actually easier for everybody to access if in case people have missed out.
I'm just dropping. So this is the website that you can just go to Google and search metascape.org. Any one of you, can anyone from the team just do that?
Yes, I'm asking others to do that. Yeah.
You can ask. This is an interactive session, so please do ask if you fail to follow any of the steps. So I will be waiting until most of you are on this page if you have the data set. So meanwhile, if you don't have the data set with you, you can just, I can browse you through the data set. As I was telling that you would see there is a lot of variation. So this is from a recently published publication from our lab. We did analysis of the swab, nasopharyngeal swab proteome. Now, I think if you were following Dr. Debodip Bhattacharya's lecture, he also showed that how powerful mass spectrometry-based studies could be. So what we took was simple swab, nasopharyngeal swab of patients, and then we did a mass spectrometry analysis. Now, all these patients were primarily being diagnosed at a very early stage of the disease. But how do we know that how the disease pathways are altered? How mass spectrometry is a really wonderful tool, and how network analysis can help us understand the disease. So these were the genes that were differentially regulated. If you can see my screen, if I go down, you would see this is fold change, positive versus negative, and there is the t-test value. So these are log fold change values. So you can see these are the few sets of genes. The yellow highlighted ones are highly overexpressed. Quite a huge number of genes that got overexpressed. And then there are a few genes that were Can you guys follow me? Hello?
Your voice was slightly breaking in the last sentence. You can repeat again.
Okay, so what I was saying was if you are having the Excel sheet with you, so these were the mass spectrometry values that we got for individual proteins. So these are the abundance values. And then we did analysis of the fold change, like how much did it, whether they were upregulated or downregulated. We also conducted a t-test to check if they were statistically significant. This is completely curated data, which I told you that we cannot just push in any data set that you can do for just understanding how the software works. But once you have your experimental data, which is your own data, please do take these steps, follow the steps. You have to do the data cleanup pretty properly, and you cannot engage in, because if you throw in any wrong data set, the results will be coming all wrong. So these are the genes that were upregulated, and these were the genes that were downregulated, the ones highlighted in green. Now, if I want to do we do want to do an enrichment study and understand which were the pathways primarily altered by the disease, COVID, that also is a very primary at the primary stage. So we see 256 genes were actually upregulated. So we will take, what we will do is, we'll copy these entire gene set. So these are UniProt IDs. I will copy. I have opened the Metascape website. Then I will paste. So that's your first step that you need to do. Then we have to submit. Now I told you that I am taking 256 IDs, but what Metascape told me that they can only map 251 IDs.
Excuse
So
me.
see, there are 5. Hello. Yeah, please.
Yeah. Can you please explain what I have to copy?
Yeah, if you follow my screen, you see there is only one sheet.
Yeah.
First column has the UniProt ID of all the proteins.
Yes.
Okay.
Yes.
The second column has the gene name. You can copy any one of them.
Okay.
Okay.
Yeah.
But don't mix and match. Don't copy half UniProt and half gene name. Then you would have nothing in your hand.
So I should copy the entire column?
Not the entire column. You see, if you go on the extreme right hand side of the Excel sheet, you would find the FC. So this is fold change value, and this is positive versus negative, which primarily means positive COVID positive versus COVID negative patients data.
Okay.
So you see there are almost 256 genes. If you scroll down, you would see almost 256 up till cell number 256. I'm just highlighting it for you. So up till this cell, we have all the genes are upregulated.
Okay.
Okay.
Yeah.
And now we want to understand how the genes which are upregulated, what biological sense, because if I look at this Excel sheet, I really am I audible?
I lost you actually
Now you are audible.
last.
Yes,
Yeah. But I think I have understood. Thank
go
you.
ahead, please move fast, Arup. Slightly, we are moving into the lunchtime now. But I think the list of genes one could always select as per your discretion as well. Either you can go with the cutoff of the four. Anything above 2, 4 up and down, and also p-value passed. Even one could only do analysis from that. So please go ahead now.
So once I have done press submit, you would find analysis as a species. So it will ask, it has automatically detected these UniProt IDs are primarily from Homo sapiens. But there are homologs in Mus musculus and Rattus norvegicus and Daniorario and Drosophila and C. elegans. So it will also throw up these homologs. So basically, when you are analyzing your data, you should be very clear what kind of which species you are using. But one more time, I would just, if you remember in my PowerPoint, I was telling you that there is an issue of coverage, species coverage. This issue is that you won't find that there is 100% coverage. There would be few UniProt IDs or which would not map with your current database settings. This is because not all databases are not updated with all the latest versions. So all databases, these UniProt IDs we get from the MaxQuant analysis or the Proteome Discovery analysis, that the version they run might not match with the version your pathway and enrichment tool is working. What you can do is you can try to manually map, or else we can, since out of 256 you got 251 mapped, you can go ahead with that as well. So I will select Homo sapiens 251, and there's an option of express analysis and custom analysis. So we are running out of time, so we won't go into custom analysis. But what express analysis does is it uses a default setting. So when you're starting off with your data analysis and you're going to get a flavor how the data looks like, you can just click on express analysis. So if you will see, it will working on, it says working on ID conversion, working on gene enrichment. So you need the server, it's a server-based program, so it will take some time. And it also depends on your length of the input list. In enrichment studies, if you use very short input list, you won't get anything significant. That's something because your statistical power is reducing. If you just put 2 proteins, you will come up with 0 results. So basically, these are for high throughput data. Am I audible?
Yeah, you're audible. Go ahead.
Right.
So, okay, I want to ask, input as species here in your screen it is showing 251, but when I'm trying to run this, it is coming 7 only.
7? I don't know. I mean, it should be coming.
At least once you can show the whole thing, and then we can take the troubleshooting separately.
Okay, right. Thank you, sir.
So you see, it has stuck at 40%. So what I did was I did a same analysis earlier. I used the same settings and I have done this. So what the Metascape gives you as an output is something like this. So first, it will give you which are the most probable pathways that are upregulated. So you see these are the identifiers I was talking of. So anything just starts with R is the reactome gene set enrichment. So metabolism of RNA, regulated exocytosis. So these are the types of information. These are the pathways that got enriched. Now, interesting things you just need to study as we go down. You see, it is taking platelet degranulation, interleukin 12 family signaling, right? These are very trademark signals that this is a disease pathway. This cannot be platelet degranulation won't happen in a healthy patient. So these platelet. Similarly, if you just go down, then there is also a thing called the top 11 gene ontology biological processes. So this is so if you want to click on it, a heat map will come. You have to download the heat map. And here it is. So the heat map, if you see, you would see what are the biological processes that are most upregulated in case of COVID positive patients. So negative regulation of biological process, localization, metabolic process, regulation of biological process, immune system process, multi-organism processes. So which basically means there was an infection. So it's not as coming from a single organism. So these are the pathways that the software is predicting that got upregulated. If you want to see how the p values, so this is another output. So that was a pictorial output. This is in a table format. So it shows what is the gene ontology pathway, then which category it falls under. So it's a reactome gene set. Then what does it describe? It's metabolism of RNA. And then you have percentage enrichment. So percentage enrichment, how much of your data set actually matched with this particular pathway. And count is the number of genes that were involved in that pathway. So out of the 256 inputs that we put, 61 proteins were involved directly with metabolism of RNA. Then we get this wonderful depiction of the clusters. So if you just click on PDF, you will get a PDF file showing a cytoscape based output of how your pathways are looking. Cytoscape is another software that is very powerful and that can be used to visualize these pathways. So the same information that was there in a heat map format, it shows in how the proteins are interacting, which kind of pathways and networks they form. Next you get is the protein-protein interaction network. So all you need to do is again click on PDF, download, open, and you see you would get a wonderful description of how the proteins are interacting, what are the protein interactors, which are your pathways, and it will give you the M code variant. I will come to the M code. So these are the M code. So you see M code 1 is regulated exocytosis. You can go back to your data and try to see where was our M code 1. So M code 1 is in green. So this is our M code 1. So this cluster of proteins primarily interacts together and it regulates exocytosis. Then there is another cluster of proteins that is for membrane trafficking. So this M code 1 is according to gene ontology database. Then there is another M code 1 that is according to the reactome database. So since this is a meta-analysis, you would find your data is analyzed through multiple databases and all of them are represented here. So you really do not have to run to individual databases with your data set. You just click one, you submit your entire gene list, and then you would find what kind of analysis all different databases are doing. And then you have to use your biological question, that what was the question I was asking. So there should be this, alliance between your biological question asked and the kind of data analysis you want to do. Then there is the quality control analysis. I was talking of transcription factor targets. So these are the transcription factors and how many proteins or genes rather were the target of these transcription factors. So if you want to understand a transcription factor biology of your of the proteome you are studying, then this tool is very important. So you will always know these are the specific transcription factors and how the genes were upregulated based on these transcription factors. Next comes again. So if you see, these are the genes regulated by SP1, maximum genes were by SP1 transcription factor, then there are CREB, then USF1, NF kappa beta 1. So these are all, this entire analysis can be done. Then this is enrichment summary using the DisGenNet database. So these are all figures are from different databases. So there is undifferentiated carcinoma, anaplastic carcinoma. As I said, it will throw up with all kinds of, even the network analysis will give you all kinds of data answers, but you have to use your biological question in mind and then look at the data. So we were using COVID, and also it happens that what kind of data, since these are all curated databases, manually curated databases, so it also takes some time. So if we have this COVID is a new disease and it just came up now, so maybe if you repeat the same analysis and the DisGenNet server gets takes more manually curated data from COVID-19 publications, maybe 6 months later the same analysis will come up with a different result. So these are the things that you need to keep on mind. And this is the Pagene database, the Pagene base, which shows what kind of where did these proteins come from. So if you talk of tissue, most of them originate in liver. If you talk of cell species, cell specific, you see there is the bronchial epithelial cells and trachea. So you get a hint that these might be a nasopharyngeal swabs. So this is how Metascape works. And the final summary analysis is an enrichment analysis in COVID, which I did not discuss because it's something very particular to COVID. But since it is a COVID data set, the software has very smartly detected that there is a significant match between what we are reporting to what other people have already reported. So these are, omics data set of published publications from COVID-19 analysis. And you see, since ours was data from a COVID-19 base, we can see there is so much of match with already published data sets. So this RNA from Sun et al. Then there is the translatome from Bochkova et al. Then this from Stukalov et al. Papers. So this gives you this is more of a quality control, and you can again take these information and build on your discussion for the paper. So this is Metascape in general. If you have any questions, you can start asking right now.
All right, thank you very much, Dr. Arup. So this was, I'm sure, good learning for all the participants to see like how to now take your static data from Excel sheet and start making it dynamic and trying to gain some sense out of the data. What I suggest now that, all of us can take a lunch break now. We are going to meet at 2:30 again. We would like to now do these analysis by the participant themselves, which I think they have already started doing it. And if they have encountered any issue, then we can handle that in the next session, where I also want to show them a little bit about the microarray experiments in the lab. And we would also like to have some brief discussion about some group activities, right? All right, so everybody, please take lunch break, which is well deserved since morning we have not given you any break. And then let's resume back at 2:30 p.m. Arup, thank you very much for this session, and thank you everybody for staying active in the whole sessions. We are stopping this now, but we will again join back at 2:30 p.m. Okay, thank you very much.